Okay, we're back. This is the second part of this morning's discussion. This is the last day of our public health applications course. And here we're just looking to discuss a bit on the more positive side. We've talked about data, we've talked about thinking frameworks, we've talked about tools and possible analyses, things that we can learn. But what are the opportunities that open up? So I'll throw out one comment or one idea, which is appearing in the literature and in people's minds more and more. We've just shown you tools and ideas and frameworks for getting good synthetic maps that have excellent predictive ability for species. So a very logical extension of that is, why don't we just model everything? Which is to say, why don't we work through all public health relevant organisms? All the mammals, all the birds, all the mosquitoes, all the sandflies, all the ticks, all the fleas, etc., chagas bugs. Why don't we just do everything all at once, one big massive effort, and have a niche model out there in the community for everything that then the public health world could jump in, grab it, and use it? It's at least worth discussing. I have reasons why I would say maybe not right now, but I can start it off, or if anybody else has things on his or her mind, jump in.
Go ahead, Tom.
Thank you, Lindsay. I hope that we've impressed on you the need for essentially manual handling of a lot of these steps. We talked about, for example, M hypotheses. So if you've used if you've used any of the many modeling algorithms, most of them have the opportunity to allow you just to load in 100 species and develop a model for each one of the 100. So a very big temptation is just to, let's download all the data from Mosquito Map and feed all those data into Maxent and produce a model for every one of those species. But I hope by the end of this course, which is to say today, I hope you've appreciated that we need to consider each species individually and think about its accessible area and think about its area that's been sampled, and essentially develop a customized calibration area for each species. And we may need also to customize the parameterization and the calibration process for each species. Think about what I showed you with no silver bullets. We may even need to use a different algorithm for each species. So what we can't do, I know I said that this would be a more optimistic session, what we can't and shouldn't do is just model everything in an automated sense. But I think there are big opportunities to assess what parts of the medically relevant or public health relevant biodiversity world are sufficiently sampled, and then develop workflows and protocols that allow us to genuinely and robustly model each species. It might be, the 62 species of triatomine bugs in Brazil, or it might be, all of the medically relevant mosquito species in Guatemala, what have you. I think that's feasible at this point, which can then provide detailed maps of public health relevant taxa that will then indeed be more accessible to the public health community. Any thoughts from other people?
I would like to ask you a question, Stan. I'm going to give a comment to the questions, actually. Why you don't model Ebola?
Why I am not modeling Ebola? Well, I did model Ebola beginning in 2002, 2003 with colleagues at CDC. And those initial maps were published in Emerging Infectious Diseases in 2004 in January. And a very interesting happening was that the maps we published in January 2004, actually for Marburg virus, the sister genus to Ebola virus, those maps managed to anticipate the very unexpected outbreak of Marburg in Angola, which happened at the end of 2004. And I've published updates of those model predictions into 2006, 2007. Now, Ebola is very much in the public view right now. But I do need, and I think this is why you're asking me this question, yes. I do need to specify what it means in the sense that we have managed in this course, what it means to model Ebola. And that is to say, in the sense of developing these models of risk of, I'll say, outbreak, we're really most interested in only one of the, 6,000 cases that have resulted from this most recent outbreak. And that one case is the first one. There's good published evidence in Science a couple weeks ago that this outbreak came from a single initial case. That initial case is the one that is interesting as far as the ecological niche of Ebola virus as a zoonotic phenomenon. We don't have the luxury of knowing a lot about the reservoir of Ebola. So we can't, for example, go out and sample all of just that reservoir taxon across all of Africa and be able to see Ebola when it's not causing human disease. Rather, what we have to do, unfortunately, is harvest information from human case data. Indeed, I have now updated the data set that I used 10, 12 years ago, and I have what I believe is a complete compendium with geographic references and with uncertainty estimates of all instances known of transfer between the zoonotic world and the human world, or the great ape world also. So I think indeed everything's prepared for another round of modeling. Indeed, another research group has just published very recent new niche-based models for Ebola. I would argue with some of their data management practices. For example, they didn't take into account the spatial autocorrelation of some of their occurrence data, which I think are putatively non-independent. But I can easily see coming back to the Ebola question very soon. If anything, to avoid very, very broad-scale panic about this disease. It's dangerous, and that's being demonstrated in West Africa right now. But Ebola is transmitted only in a very restricted set of climate conditions. Now, when I said that only one of those 6,000 cases, probably more than that by now, is relevant to these models, there's a whole other set of approaches that could be marshaled to looking at the spread within this current outbreak in West Africa. And those models are more based on human connectivity and movement. How do people move in Liberia, Sierra Leone, Guinea, and between those countries and other countries? By air, by ship, by train, by car, walking. How do people move? How do materials move that have been near people who are sick? And if one could summarize essentially those contagion events, then one could anticipate the behavior of this current outbreak. And I know there are several research groups that have that work in progress.
Thank you, Tom.
Other thoughts from people around the virtual table here?
I just want to say that, for example, your question about why do not model all the potential reservoirs or vectors of one disease in a bunch, in a batch, something that we must to have clear, for example, is that these systems are too complex, that there are some reservoirs or some vectors that are more competent for one disease compared to others. So maybe I have two species, and one species is going to be really resistant and is going to have the potential to spread the disease more easy compared with other reservoir that is going to be, let's say, more weak and is going to transmit the disease in a very short period of time, for example. So if I model both reservoirs, maybe this one that is not very relevant for the spread of the disease is going to add some noise to the model, and it's going to show me some areas that are of interest when in fact I should take more attention for the more important reservoirs. So all the mammal species that could, or vector species that could be involved in the system of one disease are going to have different levels of impact for the spread of the disease.
That's a very good comment. In a way, I think we get at that. Remember our discussions of component-based approaches to putting together risk maps. A really good component-based approach would then weight those different reservoirs or those different vectors by their importance in the system. But of course, that depends on having information about those weights and those relative importances.
Another thing to think about when we're looking at making niche models, whether it's for the vector or for the disease at hand, is what's the goal of modeling? So we can't plug and chug and just standardize how we run niche models for 15 different types of diseases, because the goal of what we're looking at might be different. Whether it's, are we looking at the potential risk of a disease spreading or invading, if you might say, to another area that has never been before? Are you looking at renewed chances of future outbreaks? Are you trying to model past outbreaks to see where it may have come from? There are so many ways to look at it, and so each time you're making a niche model and thinking about a disease or thinking about, especially if it's a vector-borne disease, you have to really stop and be able to frame that question before you actually go in and model, because that changes the factors and the variables that you need to really focus on.
That's a very critical point. If you are interested in, what are the environmental risk factors that shape emergence of this pathogen, then you're going to develop your model very, very differently than if you're interested in the question of where is this disease most likely to be transmitted. And that will be very different if your question is, where is this disease likely to be transmitted 50 years into the future? And so each one of those would have perhaps a different set of environmental parameters, very different paths of data management, like do you use a principal components analysis to compact your environmental layers or not? So that's a very good point from Kate, that you can't just say, oh, I ran a niche model. I'm done. Nobody else needs to run that niche model for that species. It doesn't work that way. Other thoughts? I'll just throw out one more kind of topic of discussion. I've just shown you an hour ago how the biodiversity community came together to produce what is now an invaluable data resource for the biodiversity community. And we talked a little bit about how, well, maybe the public health community should take those same steps, which would greatly enable public health analysis. I wonder, and again, this is just something I'm throwing out, are there opportunities for collaboration between these realms? I mean, we have these big data realms. I could list disease occurrence data, biodiversity occurrence data, taxonomic data, geospatial data, remote sensing data, okay, geospatial data, human characteristics data, censuses, population, socioeconomic status. There are these big realms of data. And one of the big lessons that I've certainly learned over a couple decades of doing this sort of work is that those data realms are very much unlinked. They're very much separate from one another. Any thoughts about that from our virtual panel?
I just want to do a kind of call to all the people working in public health that they are maybe not interested in working with mapping diseases, to have the good manner of publishing, for example, if they are going to collect some data, serology in rodents, or I don't know, if they are doing field work, maybe it could be good idea, like add a specific location of the areas in which they have been working, because there are a lot of students that are collecting that kind of data from papers, in which paper we found one disease in one mammal, for example, the exact location. And currently we have this opportunity of sharing a lot of information using supporting information in the journal. So I guess that could be a good start to share our data if we have a lot of data in the desk, and maybe we are not going to use it.
That's a very good point, Luis. When I guess I'm a little extremist as far as data sharing. In general, for me, if I go out into the field and collect data, those data are online and available to everybody as soon as those data are digital. But I am an extremist, I admit it. I don't really expect the whole community just to put all of their work in progress, all of the data online for everybody. Certainly when we're talking about students or young faculty, getting that publication may make or break your career. But I would put to everybody that the vast bulk of public health relevant data and the vast bulk of biodiversity relevant data is not in use right now. Data that were collected for a study 10 years ago, data where the paper already came out, data where the researcher is retiring, data where the researcher has moved on, data where the outbreak is over, and yet those data stay in our file cabinets or in our computer hard disks, and they just sit there. And the terrible thing is sometimes they disappear. You know, if I have a heart attack and die next week, any data that I haven't shared are probably poorly documented and probably none of my students, including the 3 or 4 here present, is going to take the time to go through the hard disk and figure out what those data were and put those data in, thank you, Kate, put those data into the right format and get them out there. So it really involves a cultural shift where, first of all, data are never just for one research project. Data are not temporary. Data are permanent. 2 is you never think my data. Okay? You think, well, I've got this data set that I collected, and those data are a common science good. Right? They are a commodity that has value to science, but they're never yours. They may be lent to you for a while. You may have improved those data such that you are going to get a very nice publication out of it or make a very impressive report to your boss. But those data are not yours. Those data are everyone's, or should be. And then maybe a third consideration about data should be that everything that is done about a data set should be archival. So if we were to go a kilometer that direction to the Spencer Research Library here at the University of Kansas, they are a collection of beautiful manuscripts where literally one copy exists in the world. Everything is archival. The folders in which a manuscript are kept are acid-free. There's no light that ever gets to the materials of interest. They're inside boxes and inside cabinets. Everything is done with an eye to those materials being in exactly the same state as they are now 100 or 200 years into the future. Or if we go 2 floors up in this museum to the bird collection, there are 120,000 bird specimens sitting there, and every one of them is sitting on acid-free media, in an acid-free environment, in an environment that's free of ultraviolet light, in an environment that's free of insect pests, with humidity controlled to be low, etc., etc. And again, everything is about those specimens existing and having the same utility for science in 100 or 200 years. We should do the exact same thing with our data. That would change the way we work with our data. We have right now a culture of temporariness with respect to data. Every single data point, when you add a georeference to it, that georeference should be transmitted back to the, I want to call it, owner of the data point. It might be a specimen in a museum in South Africa, or it might be a data record in a database in somewhere else. Now, right now, there aren't good mechanisms for reporting those data back. I have a project on Birds of the World where I ended up correcting about 8,000 scientific names. They were non-standard names, but it was possible to figure out what they should be. And the only way I could report those corrections back to the data owners was by emails. That's not efficient. So, again, trying to be positive, I think there are big opportunities for collaboration, and there are big opportunities for major payoffs in terms of learning. To me, one of the biggest realms of opportunity is if you can manage to link up those very disparate data realms, and that would make a big difference.
I'll make a comment to that.
Of course.
A big step with that, once you get everybody on board with sharing data and collaborating and working together, which is a big enough task as it is, the next step is standardization. How do you standardize the data so an ecologist can work with disease data or work with spatial data. A good case in point is LiDAR. So LiDAR has been around for, I don't know, decades now, and it's still not standardized as to how you submit the metadata for these LiDAR data that you collected. So if I, as an ecologist, want to do some modeling utilizing LiDAR data, I have to go through each and every data set going, okay, they've recorded it this way, I have no idea what wavelengths they used, where did they store it, what formats it in, how did they clean it. And it's the same thing with biodiversity data, it's going to be the same thing with the disease data. We've got to find a way to standardize it so everybody can access it and know they're getting the same quality of data.
So that takes us back to the issue of common vocabularies.
Yes.
So a very early lesson in the biodiversity world was that we will not be able to standardize how 100 institutions manage their, let's say, natural history museum specimen data. It's not going to happen. Okay, it's like the proverbial herding cats. So what was done in the biodiversity world was this Darwin Core, where it is an abstraction of any one biodiversity database format, which is to say one given collection might save its information in terms of day, month, and year. And another biodiversity data set may have one of these synthetic date formats, where essentially day, month, and year are coded together. There's no way that we will convince the Smithsonian Institution and the Paris Museum and the University of Kansas all to use the same date format. But the Darwin Core specifies here is how you report a date in Darwin Core. And so it's actually pretty easy to take the University of Kansas data set and translate those data into the Darwin Core format, and take the Smithsonian data set and translate those data into the Darwin Core format. So it's kind of a meta-standardization. And the same could happen in other data realms. I think, personally, that for public health relevant data, for disease case data, that would be a major step forward. It won't be easy. It wasn't easy in the biodiversity world either. Okay, so from Andrew Elo, who is a veteran of an earlier BITC course in Kenya in 2013. Andrew, he's from Abuja, Nigeria. Andrew sends an email asking, how can we collect public health data for modeling when data collection here in Nigeria might not be accurate? And that's a very good question, Andrew. And it's not just Nigeria. Not only might the data in Nigeria or any other country not be accurate, but the data collection might have inherent biases. The data frequently, or almost universally, won't have information about precision. And sometimes even you get cases in which political considerations come in. For example, a country might not want to admit that it has these public health problems that allow diseases to emerge. And so there may be political considerations that come into essentially purposefully biasing the data. So how do we get around that? I would suggest, very generally, some guidelines. Multiple independent data sources, and testing for consistency among those data sources. Essentially, exploring the data in ways that may allow you to detect biases or problems within any one of your data sets. So for example, we just talked about health map and extracting data from ProMed posts. And that has its problems. But if those data provide us with essentially a profile of a disease, we can compare those to data from perhaps within a country. You brought up the example of Nigeria. So we could look at disease occurrence for whatever disease across West Africa and ask whether there is good consistency between the data for West Africa and the finer scale data within Nigeria. If they disagree, then we have to think about why. But think about the maps that Lindsay presented yesterday for Buruli ulcer. We could take those West Africa-wide maps and compare those to the data existing within any of those countries, data that are independent, and we could ask whether we're getting the same signal. Thoughts from anybody else?
So I have a couple of thoughts about this, and it's a little bit more general than what Town was talking about. But I think that it would be, I would like to see a little more emphasis on data collection from larger research, from research grants, and having a stronger emphasis on the actual infrastructure, building infrastructure for data collection. I think that a lot of times people go and get the data and then they kind of leave. There's no infrastructure available for people to continue putting these data points into kind of a standardized format. And also there needs to be more emphasis just on following these research questions through all the way in the locations where people are collecting data. So that way there's actually a full infrastructure continuing to build across the entire globe, and that'll make it much easier in the long run for people living in locations where there's tropical diseases or other vector-borne diseases to then upload these data and build these infrastructures themselves. So in the one hand, I think that there's definitely ways to try to look at data across different regions to see if you have a bias in it. But it would be nice to see just an overall stronger emphasis on data accuracy and collection from grants like NIH or NSF here in the US or other countries.
That's a very good point. The US National Science Foundation has recently emphasized points along those lines by every time one submits a proposal to the NSF, one has to provide a data management plan. And that is literally a documentation of how you will manage your data. Some of it's very proximate. Some of it is how do you avoid lab disasters in which you lose all your data 4 years into the project. But some of the data management plan is how are you going to make those data available to the community, assure their long-term survival, document them appropriately, and that data management plan actually now participates in making your proposal successful or not. So that's very crucial. Another point I would make, essentially another avenue towards promoting these ideas, is think about how genomic data are managed and saved and preserved. If you want to publish genomic or gene sequence data in a paper, almost every journal requires you to provide the GenBank accession numbers. And you can literally just go in, copy that GenBank accession number, search it on Google, and get right to a digital object, which is the sequence data and the associated metadata. And there's been some explorations in the biodiversity world of what would happen if scientific journals required deposition of biodiversity data. And not just, here's a PDF, or here's an image of a table, or here's a Word document with an undocumented table. I'm talking about Darwin Core standard format biodiversity data sets. And that's happening now. The journal Zookeys is experimenting with this. And we have a beautiful example from the gene sequence community. Read molecular biology or molecular phylogenetics and evolution, and you'll see that essentially every publication has to document and deposit its data set at GenBank. So there are avenues, there are examples that are out there as far as how one might proceed along the lines that Lindsay is suggesting. I agree 100% that working with what has been called found data, like data you found along the way, is a bad idea. And much better would be to be able to go to more authoritative data. As Abdallah said, documenting not just the presences, but also where the studies were done so that you can reconstruct the absences as well. And that can enrich models quite a bit. Other thoughts from anybody?
Tom, I need to add a comment actually for the questions, because he asked about how to collect the public health data and how to assign an accuracy. The system mostly work in Africa.
Are you there?
Yes.
Keep going. I'm listening, and everybody's listening.
Okay, so the system work in Africa, like each village have a ministry health office or ministry health unit, and afterward they will update the data and send it again for higher authorities. And if he went for the high authorities like governorate or county or state, according to the system work in Nigeria, he can get all of these data, and afterward he will begin to work with georeferencing of these data by returning back to the data again and to each village which is sampled, and afterward he can confirm what is the georeference for each case, which is very difficult strategy. But if you need to look for Africa, for example, on GeneBank, you will find that the data available for DNA sequence is more than the data available for disease or georeferenced disease cases. And this actually, if GeneBank is going to assign like a requirement which is urgently must be submitted for each sequence that's submitted for GeneBank to assign a longitude and latitude, this will be actually great advancement or great progress for GeneBank database.
Thanks, Abdallah. Yes. Got a response from Andrew Elo, essentially pursuing these questions further. Thank you, Andrew. What he asks is, so if disease data are not there, and there's a possibility of using vector data or host data, what do we do for diseases that are not transmitted by vectors? So, for example, coccidiomycosis, it's purely environmental. If we are lacking the public health data, and if we're lacking any animal agents that could help us along, what do we do? I think certainly that's a bad position to be in, Andrew. But there have been quite a number of efforts that take what is known of disease etiology and in a GIS then develop essentially guesses at risk based on that etiology. So, for example, there's a whole suite of diseases that are held in soil. These are the coccidiomycoses, held in soil and transmitted directly to humans, essentially via respiration. And there have been efforts to model risk for diseases like that, simply based on where are certain soil types present, where are certain atmospheric conditions present, and perhaps where is the soil surface being broken, plowing, construction, things like that. That doesn't make for a very easy situation. And always, when you do something like that, what you're going to want to do is find some way to test how reasonable those predictions are. They may be completely unreasonable and completely useless. And that goes even when you do have the data on the cases or on the hosts or the vectors. If there's no model evaluation in there, don't believe it. Okay? If there's no evaluation in there, it better be couched as, this is an exploration under these assumptions. But you can imagine developing a GIS-based exploration like I just described for the region you're interested in and for another region where maybe there are data. And if the region where there are data in existence, if your risk mapping approach works, then maybe you can use it in your country where there aren't data.
So I think a good example of this is brutally ulcer disease, because although we have some data on locations of people who've had brutally ulcer disease, we don't necessarily have any information about the ecology of the bacterium or the exact time frame that a person became infected with the bacterium, because we don't know how it's transmitted to people, and each individual lag time of when they get the disease and then another lag time of when they go and seek medical treatment is different. So, for example, with the projection of the niche model that I showed yesterday in the talk, I think that I kind of touched on talking about looking at something like that as a first tool to then go ahead and go in and look at areas more specifically and maybe use these projections as a way to narrow down potential study sites for research, for environmental sampling, or for people to go ahead and look and see if there are elevated numbers of cases in certain regions. But it's certainly not, in that case, a risk map in the sense of this is where transmission is going to take place. So it is possible to explore different environmental variables or ecological variables related to a disease. But then, as Town was saying, you absolutely have to follow up and see whether or not your model is corresponding with what's actually taking place. Mm-hmm.
Okay, let's go ahead and wrap up this session, and then we'll come back for a final session on kind of getting back to the question of public health and biodiversity. So see you in 5 minutes, everybody.
Bye-bye.